Okay, good evening, students. Okay, good evening. We have been discussing. Good evening. We have been discussing about signal transduction, and as part of this, we are also learning the mechanism of hormone action, both together. And coming to signal transduction, we have seen three different types of receptors. One, ion channel linked receptors, second one G protein linked receptors, and third one enzyme linked receptors. And out of these three receptors, we have already discussed about, in addition to this, two different types of ligands, one that can enter the cell, one that cannot enter the cell. When we see the hormones, three types again, just to refresh your brain. There are three types of hormones based on chemical nature. Some are proteins or peptides, some are steroidal, and third one are derivatives of amino acid called as tyrosine. When we compare these three, proteins are hydrophilic, large in size, so that they cannot enter the cell and they remain outside the plasma membrane. So these ligands, which are proteins, if it is a hormone or anything, it has to bind to a receptor present on the cell surface, and its function is mediated by a second messenger. And we have discussed about second messengers like cyclic AMP. Okay, and the second one is steroidal hormone. So this steroidal hormone, because it is small and lipid soluble, lipophilic, it can pass through and it binds to a receptor present in the cytoplasm. If this receptor is an enzyme linked receptor, the activity with respect to the target cell within the cytoplasm is performed. If it is a hormone which is said to be derivatives of tyrosine, here this ligand or hormone not only process the cytoplasm, it can also enter the nucleoplasm where it binds to a nucleoplasmic receptor. And the hormone receptor complex then binds with the DNA to the specific gene where there is a promoter. That promoter is called as hormone response element. Then what happens? Here, as soon as the ligand receptor complex binds to the hormone receptor element, transcription of the target gene takes place, followed by translation, production of a protein, and this protein that is present in the cytoplasm now is responsible for what we call performing a uh, uh, cellular response within the cell. So these three types we have seen, and these are said to be mediated by a number of signal transducers. One such is said to be protein kinase A. So in short, we can call it as A kinase or PKA. And this PKA is CAMP dependent, cyclic AMP dependent protein kinase A, which acts as a signal transducer. We know that cyclic AMP exerts its effect in animal cell mainly by activating the here enzyme CAP dependent protein kinase A. We also know what kinase is. Can anybody tell me what is the function of a kinase? Excellent. It is responsible for transfer of a phosphate group so that phosphorylation of the target protein takes place and this phosphorylated protein gets activated. So here also the same thing. Protein kinase A is involved in the transfer of the terminal phosphate group from ATP. I already told you there are three phosphate groups in ATP, and this terminal phosphate group is the energy rich. Okay, and this terminal phosphate group of ATP is transferred to the target proteins. So let us see, there is a target protein with a number of amino acids, and there are so many amino acids, phenylalanine, methionine, and so many such kinds of amino acids are present. But here, the terminal phosphate of ATP group is transferred to only two specific amino acids. One is said to be serine, the other is said to be threonine. Why only to serine and threonine? Why not to the other 18 different kinds of amino acids? Why? Because serine and threonine are hydroxyl amino acids. They are hydroxyl amino acids. For this amino acid, there is hydroxyl group, and this amino acid is also having hydroxyl group. In addition to this, there is another hydroxyl, another amino acid with hydroxyl group, that is tyrosine. So these three are called as hydroxyl amino acids, from which OH can be easily replaced with phosphate. That is the reason why this PKA catalyzes the transfer of the terminal phosphate to the target protein, where the serine and threonine residues are present. This phosphorylation of these appropriate amino acids, those that can be easily phosphorylated, in turn regulates the activity of the target protein. So once the target protein is phosphorylated, that target protein gets activated and a metabolic activity takes place. And coming to the structure of this particular thing, so this is taken from Google only, and here it has two loops. One is said to be phosphate binding loop, the other is said to be catalytic loop. So this one is the catalytic loop, this is the phosphate binding loop. Okay, and there is an active site that is extending between these two. The active site of this protein kinase extends between phosphate binding loop and the catalytic loop. And the ATP, which donates a phosphate group, and the peptide to be phosphorylated, that means the protein and the ATP, they are held in the active site. So here the ATP is held and the target protein is said to be also held here. So that is, it makes easier for this particular what we call kinase enzyme to transfer the phosphate group to the protein easily. That is how it performs the function of phosphorylation with an active site that is present between phosphate binding loop and the catalytic loop. As the name implies, phosphate binding loop. So here it binds to ATP. The catalytic loop, it transfers the phosphate from ATP to the protein. So both are present between these two only, and this forms what we call the active site. Okay, and coming to the uh, further reading of this, in the inactive state, protein kinase A consists of four subunits. It consists of four subunits, and two of these subunits are called as catalytic subunits, and two of these are called as regulatory subunits. So these are catalytic and these are regulatory. Each are two in number, of which the catalytic subunits, they bind with cyclic AMP. Okay, we already have seen that the protein kinase A is cyclic AMP dependent. So here, for it to be get activated, the regulatory units must be bound to cyclic AMP. The binding of cyclic AMP alters the shape of the regulatory subunits. These two subunits are said to be altered with respect to their change, causing them to dissociate from the complex. So as soon as cyclic AMP combines with them, as soon as cyclic AMP combines with them, there occurs a change in the shape of the regulatory subunits, so far which are bound to the catalytic subunits, now they are said to be separated. Now the released this, so what happens? The regulatory subunits along with cyclic AMP are separated. Now what is there here? The catalytic subunit. What is a catalytic subunit? As the name implies, it is the enzymatic part. So the released catalytic subunits are now activated. That means this part is said to be making it inactive. And this activated catalytic subunits of the protein kinase A is now involved in phosphorylation of the substrate protein molecule, and which this phosphorylation, either this is the target protein where on serine it gets phosphorylated, on threonine it gets phosphorylated. Once phosphorylated, this protein can act as an enzyme, or even it can stimulate the transcriptional activity of specific genes. Somebody please can you mute your mic? Okay, thank you. Oppa. Lavanya, please. Sharmila. Sharmila. Okay. The binding of cyclic AMP to the regulatory subunits, as we have just seen, it brings about a change in the conformation of the regulatory subunits, and they dissociate from the catalytic subunits. Thereby, the kinase activity of the catalytic subunits is activated, and the release of catalytic subunits requires the binding of more than two cyclic AMP molecule to the regulatory subunits in the tetramer. So here there are two catalytic subunits which are bound to two regulatory subunits, and now the catalytic subunits have to be removed, for which the regulatory subunits have to be bound to cyclic AMP molecules. How many? More than two. So at least more than three cyclic AMP molecules are required to bind with this tetramer, so that the regulatory subunits along with the cyclic AMP get separated from the catalytic subunits, so that the catalytic subunits are separated and they are involved in phosphorylation. Okay, next, coming to the mammalian cells. Mammalian cells have at least two types of these protein A kinases. Type 1 is found in cytoplasm. So if this is the cell, type 1 is said to be found in the cytoplasm, whereas type 2 is bound via its regulatory subunit and special anchoring proteins to the plasma membrane. So here what is happening is, let's say this is the cell. There is a catalytic subunit and regulatory subunit. So these are the regulatory subunits. Along with the regulatory subunits, there are special proteins. These proteins are called as anchoring proteins. So along with the regulatory subunits and anchoring proteins, this particular type 2 is bound to the plasma membrane and also nuclear membrane, mitochondrial outer membrane, and microtubules. So that is the basic difference between these two. Type 1 is present in the cytoplasm, whereas type 2 is bound to the plasma membrane via the regulatory subunit and the specialized proteins which anchor the regulate this particular entire tetramer to the plasma membrane or other membranes like the around the nucleus and mitochondria. and even microtubules. In all these cases, once the catalytic subunits are separated, how they are separated? When the regulatory subunits are bound to the cyclic AMP, more than two cyclic AMP molecules bind to the regulatory subunit so that catalytic subunits are separated and they become active. They can migrate into the nucleus where they can phosphorylate gene regulatory proteins while the regulatory subunits remain in the cytoplasm. So, what is happening here? This particular subunit, this is the anchor protein. If this is the nuclear membrane and this is the plasma membrane, so along with the anchoring proteins, the regulatory proteins and the catalytic protein, sorry, I just wrongly mentioned it. So this is the nuclear membrane. So to which the catalytic subunits are said to be present and the regulatory subunits and the anchoring subunits are said to be attached either to the plasma membrane or the same complex is attached to the nuclear membrane. In such case, what happens when regulatory units combine with cyclic AMP? These two get separated along with cyclic AMP. Now, what is remaining here? The catalytic subunit. This catalytic subunit is now involved in what we call phosphorylation of gene regulatory proteins. Phosphorylation of gene regulatory proteins. So that what happens? It is involved in regulation of gene expression that we are discussing from the past two classes. Some responses mediated by cyclic AMP are found to be rapid and some are said to be slow. So, response of cyclic AMP can be rapid as well as slow. For example, in skeletal muscle. So, what is a skeletal muscle? The function of the skeletal muscle is to contract. For contraction, it requires energy. For energy, it requires continuous supply of glucose. So, how the continuous supply of glucose takes place? We store glucose in two places in the form of glycogen, one in the liver and the other in the skeletal muscle. So, here skeletal muscle is having large stores of glycogen. So here what is happening is in skeletal muscles, activated protein kinase A phosphorylates enzymes involved in glycogen metabolism so that it breaks down glycogen to glucose so that what is enhanced here? Glycogenolysis is said to be enhanced. At the same time, it inhibits glycogen synthesis. If glycogen synthesis happens, what is happening? Glucose is not available. So, glycogen synthesis is inhibited and glycogenolysis is said to be increased. Thereby, the amount of glucose available to the muscle cell increases within seconds. So, such cyclic AMP actions are said to be called as rapid. In other cases, for example, growth. So, for growth need not be rapid. All of a sudden, when you are born, you are not going to grow to all of a sudden 3 feet or 4 feet in 1 year. Why? Because the response of this cyclic AMP mediated by this cyclic AMP are said to be very slow. So here these take sometimes hours to develop fully and involve changes in transcription of specific genes. For example, in cells that secrete the peptide hormone somatostatin, cyclic AMP activates the gene that encodes this hormone, and this hormone is said to be having very slow activity with respect to growth. So here, all the responses mediated by cyclic AMP are not always rapid. Depending upon the metabolic need of the cell, they can be rapid as well as they can be slow. And here comes a regulatory unit. The regulatory region of the somatostatin gene, regulatory unit of the somatostatin gene contains a short DNA sequence, and this we call it in short as CRE. So you have already heard this about this HRE. Can anybody tell me what HRE is? Excellent. Where is it present? Excellent. So it is nothing but the promoter region of the DNA. So that from the at the promoter, RNA polymerase binds, and this gene is get to be transcribed to form the mRNA. Likewise, there is another such element which acts as the regulatory region of somatostatin gene, and this is called as cyclic AMP response element. That is hormone response element where the hormone receptor complex binds there. Here cyclic AMP binds here, so it is a cyclic AMP response element, and it is also present in not only somatostatin gene. The same CRE is also found in many other genes that are activated by cyclic AMP. Next, a specific gene regulatory protein called CRE binding protein recognizes this sequence. So this CRE is recognized by another sequence called as CREB. Okay, so the CRE has to be recognized. A specific gene regulatory protein called CRE binding protein recognizes this sequence. So there is a protein, that protein can recognize this promoter which is called as CRE here. When CREB is phosphorylated, so this protein, which is the regulatory protein, now it gets phosphorylated by the protein kinase activity. So when CREB is phosphorylated by protein kinase, where in general either serine or threonine, but here with respect to somatostatin, it is phosphorylated on just one serine amino acid. One just one just one serine amino acid is said to be phosphorylated. When it gets phosphorylated, there adds another one that is called as a crab binding protein, CBP, and this is called as transcriptional coactivator. So for this gene to be transcribed to form mRNA, what is required? A coactivator is required. This is called as crab binding protein, in short CBP. So three are recruited here. So there is a CRE, that means cyclic AMP response element, that is the promoter region, and this is recognized by a protein that is called as CREB, and this CREB is phosphorylated by protein kinase. After phosphorylation, one more thing adds. What is that? A transcriptional coactivator. This is called as CREB binding protein. Because it is binding to CREB, it is called as CREB binding protein. So there is a there is a CREB binding protein. There is a cyclic AMP response element. Now what happens is this entire thing stimulates the transcription of these genes. If at all, if this serine is mutated, if the serine is mutated, CREB cannot be phosphorylated. When CREB cannot be phosphorylated, this transcriptional coactivator cannot be added. It cannot be recruited. So when this coactivator cannot be added, transcription will not takes place. So it no longer stimulates gene transcription in response to a rise in cyclic AMP levels. So whenever there is a rise in cyclic AMP levels, there is every chance that transcription of the target gene to mRNA also increases. But when the serine that is present in the CREB protein is mutated, even though cyclic AMP levels are rising, no gene transcription will take place. So we can, based on this, we can simply say that the phosphorylation of single serine molecule in CRAB by PKA is the primary requisite for the transcription of these genes to take place. So let me show this with this diagram. So this is prepared by me only. So here there is a signal molecule. Signal molecule means ligand. Here what is this ligand? This is a hormone. And there is a receptor, the green colored one. What is this receptor? This receptor is said to be multipass protein. So this multipart protein is having an extracellular domain and intracellular domain. So to the extracellular domain, the signal molecule binds. So once the signal molecule binds to the extracellular protein, it gets activated, and there forms a ligand receptor complex is formed. The ligand receptor complex brings about a conformational change in the receptor. It brings about a conformational change in the receptor. And this conformational change in the receptor, now what happened? Ligand receptor complex is formed. Conformational change in receptor occurs, and this receptor now extends to the GTP binding receptor. Hence, we can call this receptor as GPCR. What is GPCR? What is GPCR? No, it's not complex. Excellent. G protein coupled receptor. So this green colored one receptor is a G protein coupled receptor because it binds to G protein. So now it bound to Z protein. Okay. So what is the chemical nature of G protein? It's made up of how many subunits? Three subunits. Three subunits: alpha, beta, gamma. Hence it is called as heterotrimeric. So now we are concerned with alpha subunit, which can exist either stimulatory or inhibitory, and we are concerned with the protein synthesis here. That means it must be stimulatory. So in this G protein, the alpha S subunit is said to be activated. So ligand receptor complex, once it is formed, it changes the conformation of the G protein coupled receptor so that the G protein gets activated. What gets activated? The alpha of GS, alpha subunit of GS gets activated. So what is happening now? Alpha when gets activated, it is bound to GTP and it is separated from beta and gamma. And this activated alpha S GS subunit now activates the next one, that is another intracellular protein, another integral protein called as adenylyl cyclase, which is also present in the cytoplasm. So now adenylyl cyclase is activated. So first step is formation of ligand receptor complex. Second step is conformational change in the G protein coupled receptor. Third one is activation of alpha subunit, and now it leads to the activation of another protein present in the plasma membrane, and this acts as an enzyme called as adenylyl cyclase. And adenylyl cyclase is now involved in, as usual, converting the ATP to cyclic AMP. So adenylyl cyclase now converts ATP to cyclic AMP. At the same time, till that time, in the cytoplasm, there is protein kinase A, which is inactive with two domains here. So what happened here? The the domain which makes it inactive is separated. How it's separated? By the binding of the cyclic AMP molecules to the protein kinase A at four different binding sites. So these are the four different binding sites. So when the cyclic AMP molecule binds to these different binding sites, this molecule is separated, and this blue one that is shown here is the activated protein kinase. So protein kinase gets activated. Okay, this protein kinase now passes from the cytoplasm. All these events are taking place from the plasma membrane up to the nuclear membrane within the cytoplasm. And in the from the cytoplasm, the activated protein kinase A passes through the nuclear pore and reaches the nucleoplasm. And in the nucleoplasm, there is another protein, that is CREB. What is CREB? CREB is CRE binding protein. It is a CRE binding protein. So this CRE binding protein, now what is happening? It gets phosphorylated at single serine residues. It is phosphorylated at single serine residue. So once it gets phosphorylated, it recruits another protein that is called as Krebs binding protein. So it recruits CBP, Krebs binding protein, and this Krebs binding protein, this entire unit binds to CRE. This is what cyclic AMP response element is, or Krebs binding element is. This is nothing but the regulatory region of this particular gene, and we can call it as promoter. Now what happens? The target gene is activated and mRNA is produced, followed by protein synthesis, and that brings about a metabolic change in the cell. So this is how this entire protein kinase activity works. Clear? Any doubt? So first step, who is going to tell me what is the first step here? Formation of, yes, good. First thing is ligand receptor complex is formed. Then what kind of receptor is it? So now this receptor is activated. That means its conformation changes and it gets activated, and it is coupled to what? It is coupled to Z protein. Very good. Z protein. As soon as it is coupled with G protein, here alpha subunit gets separated from beta gamma subunit. So we say this is activated. So activated GS alpha subunit. And this activated GS alpha subunit, how it gets activated? Previously it is inactive where it is bound to GDP. Now it is bound to GTP. And GTP is hydrolyzed, releasing GTP and GDP and inorganic phosphate. And this inorganic phosphate binds to adenylyl cyclase, and cyclase is said to be activated. So next step is activation of adenylyl cyclase. So once activated, adenylyl cyclase breaks down ATP to release cyclic AMP. And now this cyclic AMP is involved in activation of protein kinase A, which is inactive, and this protein kinase A is activated. So all these events are taking place inside the cytoplasm. Now its main function is not to directly regulate the metabolism, rather to regulate the gene expression so that protein is synthesized. That proteins are single protein, or can be numerous proteins. They are involved in regulation of a metabolic activity. And now this activated protein kinase A moves to the nuclear membrane, moves to the nucleus. So in the nucleus there is a gene. This is the gene. So this gene has CRE. What is CRE? Cyclic AMP response element. So this is the promoter region or regulatory region, we can call it so. And beside which there is a long region, and this is the target gene. So this gene is required to be transcribed to produce mRNA. So here activated target gene that is shown here is this one. This has to be activated. This has to be transcribed to produce mRNA. So for which the CRE, the promoter region, has to be activated. In order to activate this promoter region, there is required two things. One is CBP and the other is CREB. So what is CREB? CREB is an inactive Krebs binding protein. So here there is CRE, promoter region, followed by a target gene which needs to be transcribed, to which this inactive gene has to be added. Because it is inactive, it cannot be added. Now what happens is this inactive one is phosphorylated at one serine residue. It is phosphorylated at one serine residue. When it gets phosphorylated at one serine residue, now what happens? It adds another one that is called as CBP, Krebs binding protein. What is Krebs binding protein? It is a transcriptional co-activator. Transcriptional co-activator. Now this entire complex binds to CRE. This complex consists of a phosphorylated Krebs CRE binding protein and a co-activator of transcription, that is CBP, Krebs binding protein. So this complex, so what is this complex is made up of? This complex is made up of a Krebs associated with phosphorylation associated with a cofactor. This is bound to CRE. Now what happens? The promoter region is said to be activated. So the target gene is activated and the target gene is transcribed to produce mRNA, followed by translation to produce a protein. That is how it takes place. Clear? You have to read it one or two times. Then only you can understand it. Can I move to the next one? Okay. Next one. Role of calcium ions as second messenger. I have given you a list of second messengers in the morning session. Can anyone tell me what are those second messengers list? First one I am telling you cyclic AMP. What is the second one? Huh? లిస్ట్ క్లాస్లో చెప్పానమ్మా. నేను ఇంకా పెట్టలేదు, నాకు తెలుసు. క్లాస్లో చెప్పాను, నువ్వు క్లాస్లో ఎంతవరకు విన్నావని అడుగుతున్నాను. ఫస్ట్ వన్ ఈజ్ సైక్లిక్ ఏఎంపీ, second one is cyclic GMP, cyclic adenosine monophosphate, which is derived from derived by the breakdown of ATP, and cyclic GMP derived by the breakdown of dash GTP. And the third one is calcium ions. Fourth one is inositol trisphosphate, which is a phospholipid present within the plasma. If this is the plasma membrane, there are so many phospholipid molecules, and one such phospholipid molecule is IP3. When it is broken down, it produces so many, and one such is said to be diacyl glycerol. And when it is broken down, it produces another one that is called as arachidonic acid. Okay, so all these are the different second messengers. And one second we have already dealt with cyclic AMP. The same is the case with the cyclic GMP. That is not we are touching here now. Now we are going for the remaining 3 4 5 6. So let's start with calcium ions. So role of calcium ions as second messenger. Why calcium ions as second messenger required? Because when do you require a second messenger? Can anyone tell me? When do you require a second messenger? When the ligand is a protein. You have to remember this. I've told you already. There are three kinds of hormones. One is protein hormone. Second one is steroid hormone. Third one is biological amine. Steroid hormone and biological amine can enter the cell. So they are recognized by cytoplasmic or nucleoplasmic receptors. Whereas growth hormone, insulin, somatostatin, parathermone, all these are said to be large proteins or peptides. They cannot enter the cell, they remain outside. And these protein hormones, they bind to receptors and they remain outside only, and this message is said to be carried forward by a second molecule to the inside. This second molecule is called as the second messenger, because hormone is called as first messenger. So here calcium acts as a second messenger to a hormone which is protein in nature. Okay. So concentration, coming to the concentration of calcium ions, how it is regulated? It is regulated in two ways. One, through the calcium channels, so that calcium channels are opened and calcium is transported inside. And the other one is an integral protein which acts as a calcium transporter. So this occurs a conformational change. It rotates through 180 degrees and calcium is said to be delivered inside. So either through calcium transporters or through calcium ion channels, calcium concentration in the cell can be regulated. And the concentration of calcium ions is kept low in cytoplasm but high in the extracellular space or even in the lumen of endoplasmic reticulum. Can anyone tell me what is the name of the smooth endoplasmic reticulum present in skeletal muscle? Sarcoplasmic reticulum. What is the function of sarcoplasmic reticulum? Very good, excellent. It is responsible for storing the calcium ions. So, in the cytoplasm, calcium ions are, the concentration of calcium ions is said to be very low, whereas it is high in extracellular fluid, that means outside the cell. Even inside the cell, they are said to be more in smooth endoplasmic reticulum. That means the lumen of the cisternae of the endoplasmic reticulum. Okay. Now let us see how this calcium second messenger is said to be delivered. There is a receptor called as IP3 receptor. So here there are three phosphates, one attached to first, second, third carbons. So it is called as inositol 145 tris. Three phosphates are there, tris phosphate. So IP3 receptor is a type of calcium ion channel. So this is the plasma membrane. Now what happens? There is a receptor and this receptor is called as IP3 receptor. And what kind of receptor it is? It is a calcium ion channel receptor present where in the plasma membrane of the cisternae of the endoplasmic reticulum. Now what happens? It is IP3 receptor. That means it requires IP3 to be bound to this receptor. So binding of IP3 to this receptors opens the calcium channels, allowing calcium ions to diffuse into the cytoplasm. So this receptor is present in the plasma membrane to which IP3 is bound. So when IP3 is binding to IP3 receptor, now this receptor being a calcium channel, it opens up the calcium channel and the calcium ions enter the cytoplasm. Depending upon the cell type, calcium ions either they can activate various enzymes or even they can inhibit various enzymes. So the function of the second messenger is to take up the activity of the hormone inside. So if this hormone is said to be encoding a message to inhibit a metabolic activity, this calcium is responsible for inhibition of enzymes. If this hormone is encoding a message to enhance the activity inside, so what happens? It can activate the various enzymes present. But calcium cannot do this on its own. Activation or inhibition of various enzymes cannot be done by calcium on its own. It is depending upon the cell type. Calcium can activate or inhibit various enzymes and transport system. How? By combining with certain calcium binding proteins. And these calcium binding proteins are said to be exemplified by one such called as calmodulin. So calcium ions once enter the cytoplasm through the channel, they have entered. Once they enter, they bind with another protein called as calmodulin, and these are involved in activation of a number of enzymes or inhibition of these enzymes. And how this takes place, let us see. So phosphatidyl inositol is a minor phospholipid present in cell membranes. Okay, it is a phospholipid present in cell membranes. Phosphorylation of phosphatidyl inositol leads to the synthesis of two derivatives. One is called as PIP, PIP, phosphatidyl inositol phosphate simply. The other is PIP2. So PIP, there is only one phosphate. Here there are two phosphates. Hence we call it as phosphatidyl inositol bisphosphate. So phosphatidyl inositol is, when it gets phosphorylated, it releases two derivatives. Phosphatidyl inositol phosphate or phosphatidyl inositol bisphosphate. Where they are present? They are present in the inner half of the plasma membrane, not in the outer half. They are present in the inner half of the plasma membrane. And the chain of events leading to PIP2 breakdown, it is followed by the breakdown of PIP2. It starts with the binding of the ligand. What is this ligand? Ligand is a hormone. It can be acetylcholine or antidiuretic hormone to a GPCR. So now what happens? ADH is released by posterior pituitary, and this ADH now binds to the, what we call, a G protein coupled receptor. This is followed by the breakdown of PIP2. So what happens here? The activated G protein coupled receptor stimulates the G protein. Previously we have seen G alpha stimulatory, G alpha inhibitory. Now we call this as GQ. We call this as GQ. So the activated receptor, this receptor, once it binds to the hormone, it gets activated. This G protein coupled receptor stimulates the G protein. So here there is another protein called as G protein here. So this G protein is named as GQ. So this GQ is activated, and this GQ in turn activates another enzyme called as phosphatidyl inositol specific phospholipase C. So we can simply call it as phospholipase C, but this is specific only for PI. Hence we call it as phosphatidyl inositol specific phospholipase. So the coming to the sequence of events, what is happening? PI is broken down to PIP and PIP2 when it gets phosphorylated. Okay, and now it is the turn for PIP to be broken down. So this is led by an enzyme called as phospholipase C. So how this takes place? In the cytoplasm, sorry, in the on the plasma membrane, there is a receptor. This is the G protein coupled receptor to which a hormone binds. What is this hormone? This hormone is the ADH or ACH, like neurotransmitter. So when these two are bound, it activates another G protein, because it is G protein coupled receptor. It activates another G protein called as GQ. Once it is GQ is activated, GQ activates phospholipase C, which is inactive, to phospholipase C, which is active. So we are now involved in activation of, this is involved in activation of phospholipase C, and this phospholipase C is called as phospholipase C beta. This is called as phospholipase C beta. In less than a second, this enzyme PIP2 now gets cleaved, gets broken down to generate two products. One is called as inositol trisphosphate, the other is called as diacylglycerol. So this is the ultimate product. Phospholipase C is involved in breaking down PIPT to release IP3 inositol trisphosphate and diacyl glycerol. And these two, IP3 is a second messenger, I've just told you, and diacyl glycerol also is a second messenger. So let us see what are the pathways of these two. So we'll start with IP3 pathway. So let me explain you the same thing. So phosphatidyl inositol once get its phosphorylated, it produces PIP as well as PIP2. Okay, and first of all, what happens? The ligand comes, ligand binds to this. What is this ligand? Antidiuretic hormone. It is hormone. So this receptor, what is this receptor? G protein coupled receptor. So this G protein coupled receptor gets activated. So activated G protein coupled receptor activates the nearby protein. What is this protein? G protein. What kind of G protein? GQ protein. So this GQ protein once gets activated, it breaks down GTP to GDP, and finally, what happens? There occurs a for activation of phospholipase C. So phospholipase C is now activated to produce phospholipase C beta. So this enzyme is now involved in breaking down PIP2, one to IP3, the other to diacylglycerol. So diacylglycerol is having a pathway and IP3 is having another pathway. So let us see what actually happens here. So there are different hormones that use the phospholipase C second messenger system. Just one we have seen antidiuretic hormone, that is vasopressin is antidiuretic hormone, and the others are thyrotropin releasing hormone secreted by the hypothalamus, oxytocin secreted by the posterior pituitary, parathyroid hormone secreted by the parathyroid gland, growth hormone releasing hormone, and gonadotropin releasing hormone secreted by the hypothalamus, catecholamines secreted by the adrenal medulla, and angiotensin 2, which is secreted by the vascular smooth muscle. So these are the different phospholipase C second messenger system mediated hormones. So coming to the IP3 pathway. IP3 is a small water soluble molecule capable of rapid diffusion. It leaves the plasma membrane once formed and diffuses rapidly through the cytoplasm and binds to a specific IP3 receptor located at the surface of smooth ear. So what happened here is IP3, because it is rapidly diffusible, it binds to the IP3 receptor present on the surface of the smooth endoplasmic reticulum. What we have seen, IP3 receptor is a calcium channel. Now what happens? The calcium channel opens and the calcium ions are said to be transported. This is how IP3 pathway takes place. Okay. Next, the second one is DAG pathway. DAG is a lipid molecule. DAG means diacylglycerol. It is again a lipid molecule because it is derived from phosphatidyl inositol, which is said to be a minor phospholipid. So it remains in the plasma membrane following its formation by phospholipase C beta. So DAG, where it is present? It is present in the plasma membrane itself. IP3 is present in the cytoplasm, which binds to IP3 receptor on the surface of the smooth endoplasmic reticulum. Calcium channels are to be opened and calcium ions are diffusing. Whereas DAC remains in the plasma membrane once it is formed. And it has two signaling roles. One is that it is further broken down to release arachidonic acid. So DAG can be broken down to release arachidonic acid. So what is the function of this arachidonic acid? This arachidonic acid can act as a second messenger again, or it can be used in the synthesis of specialized lipid molecules of immune system. For example, prostaglandins are there, leukotrienes are there. So prostaglandins and leukotrienes are lipid molecules which belong to the category eicosanoids. So here this arachidonic acid either can act as a second messenger or it can be used in the synthesis of prostaglandins and leukotrienes which belong to the category called as eicosanoids. Okay, now the second signaling role is that it can be broken down to arachidonic acid. If it is not broken down to arachidonic acid, this DAG, diacylglycerol, activates a crucial enzyme called as phospholipase C, as we have just seen. We call this as PKC. We call this as PKC. And this PKC now, so PKC is said to be in active form. This is said to be made active. Who is doing this? Diacylglycerol. And now we have PKC released. And what does this PKC do? It is involved in phosphorylation because it is a kinase. So phosphorylation at what places takes place? At places serine and threonine residues only. Why at serine and threonine residues only? Because here they are having hydroxyl groups that can be easily replaced by phosphate groups. And so that the selected proteins in the target cell are said to be phosphorylated, and it leads to different uh, cellular activities like cellular growth, cellular differentiation, cellular metabolism, and transcription. That is how phosphoprolipase C or protein kinase C or C kinase we can call it as involved in these activities. The initial rise in cytoplasmic calcium ions induced by IP3 is thought to alter the PKC so that it is translocated from cytosol to the cytoplasmic phase of the plasma membrane. So previously, where is it present? PKC is present within the cytoplasm. Here PKC is present. So the release of calcium ions into the cytoplasm induces the PKC towards the plasma membrane to reach the plasma membrane. So it is translocated from cytoplasm to near to the cytoplasmic phase of the plasma membrane. And there it is activated by the combination of calcium ions and DHE. And activated PKC, as we have seen, phosphorylates target proteins. This is one thing. Or it can increase transcription of specific genes by activating the DNA bound gene regulatory protein. So thus, anything as far as we have seen here, one is said to be directly activating the metabolic enzymes. Secondly, it is said to be regulating the gene expression. These are the two mechanisms by which all these second messengers are said to be functioning. In another pathway, the PKC activation leads to phosphorylation of an inhibitor protein. Thereby, it releases a cytoplasmic gene regulatory protein. It can migrate into the nucleus and stimulate the transcription of specific genes. That is how regulation of gene expression takes place. So, DAG pathway through protein kinase C either activates certain target proteins by phosphorylating them, or by phosphorylating gene regulative protein, it activates the DNA to form mRNA and finally protein, which we call it as regulation of gene expression. So these are the different pathways. And finally, we come to the end. This is the CAM kinase pathway. And this is also dependent on calcium, which is bound to calmodulin. So calcium calmodulin dependent protein kinases are called as CAM kinases, and these are involved in certain signal transduction mechanisms. So calcium ions do not bring about the responses by itself, as we have just heard. Calcium ions cannot function, cannot activate anything because calcium ion is not having any kinase activity. So because calcium is not having any kinase activity, it cannot phosphorylate in any target protein. But when it cannot activate any target protein, it cannot bring about the responses by itself. But it combines with calmodulin, another protein, so that it can bring about the responses. And this calcium calmodulin dependent protein kinase are said to be found in a number. The calmodulin is found in plants, animals, as well as a number of eukaryotic microorganism. And the calmodulin number of molecules is said to be more than 10 to the power of 7 molecules, which is almost 1% of the total proteins of the cell. And coming to this, protein kinases. Each molecule of calmodulin contains four binding sites. As we have seen previously in PKA, there are four binding sites, two catalytic sites, two regulatory sites. Likewise, here also, thus calmodulin, this is a calmodulin, it has four binding sites. So to these four binding sites, four calcium ions can be bound. So once these four calcium ions are bound to calmodulin, it brings about a response. So when calcium concentration rises in response to the stimulus, the calcium ions, bind to calmodulin, forming a calcium calmodulin complex. And it changes the conformation of the calmodulin protein and increases its love or affinity for a variety of effectors. Okay, so here even the calmodulin is not having any activity or enzyme activity for itself. So calcium calmodulin singly or put together conjugated, they have no enzyme activity, but it acts by binding to other target proteins, thereby the target proteins are their activity is altered. So how it happens? Among targets regulated by calcium bind calmodulin are various enzymes and membrane transport proteins. So calcium calmodulin, what does it do? It activates a number of enzymes. If they are inactive, they are activated, or they can activate membrane transport proteins also. So, depending upon the cell type, the calcium calmodulin complex may bind to what we call a protein kinase. So, just we have seen that it is activating an enzyme. So, they bind to a protein kinase called as CAM kinase. Okay, CAM kinase. It is a cytosolic nucleotide phosphodiesterase, ion channels, and even calcium ATPase pumps, etc. So calcium calmodulin complex can bind to what? It can bind to CAM kinase. It can bind to cytosolic nucleotide phosphodiesterase. It can bind to certain ion channels, and it can bind to even calcium ATPase pump, which can transport calcium ions. So these are the four things to which this complex can bind and bringing about a response. Many effects of calcium ions are more indirect and are mediated by protein phosphorylations catalyzed by a family of these CAM kinases. These kinases, just like PKA and PKC, what they do? They phosphorylated the target proteins at serine or threonine residues. The same is the case. So what you have to remember is whether it is PKA or PKC or CAM kinase, all are said to be kinases. Their main function is phosphorylation. Where the phosphorylation takes place? At serine and threonine residues. That is what you have to remember. So that the target proteins are said to be phosphorylated and the metabolic activity is said to be performed inside. So these are the various signal transducing pathways that any question may come from any of these. So you have to remember only few things. What are the different types of ligands? What are the different types of receptors? What are the different types of signal transducing pathways? What are different types of second messengers? And what are the hormones which are using either PKA or PKC or CAM kinase or calcium ions? So these things you have to remember. I'll be sharing this entire PPT to you. You can note down the points that are required by you, and if you have any doubts you can ask me. So in addition to this, we have certain other signaling cascades. Just I will run through. These are not required for you, but even then I would like to mention few of these. One is called as jack state cascade or pathway, EGFR pathway, notch pathway, and wingless pathway. What is jack state signaling cascade? It is said to be utilized by all these different types of hormones and growth factors. So some receptors, when activated, function directly as enzymes, as we have seen, enzyme-like receptors, where the receptor itself acts as an enzyme, or are closely associated with enzymes that they are activate. So these are the enzyme linked receptors. These enzyme linked receptors are proteins that pass through the membrane only once. That means they are single pass proteins. Many peptide growth factors like all these use the enzyme linked receptors only. What are these enzyme linked receptors? These are tyrosine kinases. One example of an enzyme linked receptor is called as leptin receptor. Leptin receptor. What is leptin? Leptin is a hormone secreted by fat cells. You remember this. And it has many important functions, and most important functions of this leptin are: one is regulating appetite, your hunger, and the other one is energy balance. So leptin hormone has two functions. So how this hormone function is mediated? It is mediated by the enzyme linked receptor. So here there is a leptin receptor. So this is a plasma membrane, and leptin hormone is secreted by the fat cell. So here it comes to occupy a position of the plasma membrane where there is a receptor that can recognize it. And this we call it as a leptin receptor. So this leptin receptor belongs to a large family of receptors called as cytokine receptors. Okay. And in the case of leptin receptor, one of the signaling pathways occurs through its tyrosine kinase, and this tyrosine kinase belongs to a family called as Janus kinase family. And there is, in this family, there are so many proteins: Janus kinase 1, 2, 3, and so on. And we are concerned with Janus kinase 2. And here this signaling pathway, the ZAC, is said to be related to the tyrosine kinase, which is called as Janus kinase 2. So Janus kinase is nothing but tyrosine kinase. What is tyrosine kinase? It is an enzyme linked receptor. So hormone, when binds with enzyme linked receptor, is involved in particular activity. And this enzyme linked receptor is nothing but tyrosine kinase. And this tyrosine kinase is a great family of proteins. And one such enzyme is said to be JAK2. So the leptin receptor exists as a dimer. Leptin receptor is a dimer. It exists in two parts. Not the both are said to be connected. They are said to be two, which are said to be separated. And binding of leptin to the extracellular part of the receptor alters its conformation, enabling phosphorylation and activation of the JAK2 molecules. The activated JAK2 molecules, they then phosphorylated what residue? Tyrosine residue. Already I've told you there are three hydroxyl amino acids: serine, threonine, and tyrosine. So previously PKA or PKC or calmodulin kinase all are using either serine or threonine or both. But here this JAK protein, this tyrosine kinase, uses phosphorylation of tyrosine residue for the intracellular signaling. And this intracellular signals include what? The phosphorylation of certain specialized protein. They are called as stat proteins, where stat refers to signal transducer and activator of transcription. So see, here also there is transcription activator. This protein is a transcription activator. So that what happens? Regulation of gene expression takes place. So based on this Janus kinase receptor followed by activation of STAT receptors. This is called as JAK-STAT signaling cascade. So when we see this, so this is the leptin hormone. This is an extract from Guyton Hall Medical Physiology book. This is a leptin hormone. It binds to the leptin receptor. So leptin receptor is said to be a dimer. Though both the units monomers are not present together, they're said to be somewhat apart from each other. And these receptors belong to a family of what we call proteins called as JAK2. And JAK2 bind with what we call STAT3 binding proteins. And these STAT3 binding proteins, this is JAK2 is said to be a tyrosine kinase. This is nothing but a tyrosine kinase. So what is the function of tyrosine kinase? To phosphorylate by breaking down ATP to release ADP and inorganic phosphate. This phosphate now binds to STAT protein. So the phosphorylated STAT protein goes into the nucleus where it binds to the hormone response element, and mRNA is produced, translation takes place, and it results in a physiological effect. And the second thing is, because tyrosine kinase JAK2 is a tyrosine kinase, it can directly activate an inactive enzyme by phosphorylating it. So either activation of enzymes or regulation of gene expression. Here, this leptin, what we call JAK-STAT mechanism, is responsible for eliciting a cellular response. Second one is a Notch signaling pathway. So I have taken so much of time for you. Can you spare with me for 5 minutes, or I can share this PPT with you? You can go through it. If you have any doubts, you can ask me. What should I do? It's already 9:30. Okay. So not more than 5 minutes I'll take. Only the important points I'll tell. The Notch signaling pathway is important in the regulation of cell fate specification. Number two, proliferation and death for neuronal cells, cardiac cells, and endocrine systems, and most particularly in stem cells. For example, it determines the biological orientation of cells throughout development, allocating different cell types within a tissue. So there is a stem cell tissue. Okay, and for this stem cell tissue, this is, if it is at to be responsible for what we call, it is a bone marrow tissue. Some of these are destined to form WBC of five types. Some are for erythrocytes and some are for megakaryocytes, which lead to the formation of platelets. So here there are different allocations of these cells. One is allocated the function as WBC. The other is said to be erythrocyte. The other is said to be platelets. Likewise, the allocation of different cell types within a tissue is done by this Notch signaling pathway. So notch, where is it actually present? Notch is a heterodimer present on the cell surface with an extracellular domain and a calcium dependent interaction. So notch is said to be activated by ligand. It is activated by a ligand, and this ligand is said to be delta, or a protein belonging to certain categories called as serrate or jagged families, which you need not know. These are present on the surface of the cell, which is sending this ligand, that is called as sending cell. Okay, and let me see this completely easily first. So this is a Notch signaling pathway. So this is a sending cell. So this sending cell releases what we call delta or serrate or jagged proteins, and there on the receiving cell surface there is a notch protein, and to which what happens here, these come to occupy a position on this one. And now the notch is said to be connected to two different proteins called as Adam 10 and Adam 17, so that it increases the activity of gamma secretase, another one. And this gamma secretase gets separated, and this binds to what we call a CSL family. This is another responsible for what we call regulation of gene expression. So thus a notch is said to be involved in what we call this regulation of gene expression. So what you have to remember is, all these will not be asked in the examination. So you have to remember only this thing. Notch signaling pathway is responsible for cell fate specification, proliferation, and death for these three different kinds of endocrine systems. That is what you have to remember. And the third one is EGFR signaling pathway. Here it refers to epidermal growth factor. It binds to epidermal growth factor receptor, and it results in three functions. One is cell growth, the other is cell proliferation, and finally cell differentiation. So these three things you have to remember. And last one is wingless signaling pathway. So this is denoted as WNT. WNT refers to wingless signaling pathway, and it is a most evolutionarily conserved pathway that regulates very crucial aspects of the cell fate determination and cell migration, cell polarity, neural patterning, and organogenesis. You can see in blastula, all the cells are present outside, leaving a space inside. By the time it comes to gastrula stage, certain cells are going inside. This forms the endoderm and this forms the ectoderm. So after that, there are certain cells here which are mesodermal cells. So every cell ectoderm, these cells that are present here, they are neuroectodermal cells. And these cells that are present here are somatic ectodermal cells. Neuroectodermal cells are responsible for the formation of nervous system. So that is neural patterning, how it takes place. And certain cells from the blastula are transported inside to form endoderm. That is what cell migration is. And all these are responsible for the formation of different organs. And all these are said to be under the control of this wingless signaling pathway. Why this is called as wingless signaling pathway? It is discovered in Drosophila melanogaster with respect to the wingless characteristic first. After that, this pathway is even observed in many animals, including human beings. Hence, we call it as wingless signaling pathway. And you have to remember in what way these pathways are required. Only these names you have to remember, not the entire mechanism of action. So, with this, we conclude the mechanism of hormone action and also what we call it as signal transduction pathway. So hope you understood. So I'll share the PPT, go through it once. If you have any doubt, please post in the group. I'll try to answer it. And we'll tomorrow in the next class, I'm going to start with the pituitary gland. Okay, and all the endocrine glands will have a quick run through. So I'm telling you, tomorrow's class is pituitary gland. So if you prepare and come, we can save time. So that if you know something, I need not tell you anything, everything. So what is that is not known to you, that only will be explained by me if you come prepared. So tomorrow's topic is pituitary gland, its hormones, including the hypothalamic hormones. Thank you. Thanks for your patient listening. God bless you.